Hello everybody, my name is Hassan Nafid. I am an anatomy and physiology instructor at Madison College in Wisconsin. We will continue talking about our anatomy and physiology sessions. This time we're going to talk about one of the cellular activities. This activity is important to the cell to survive. This activity helps the cell to regenerate, to repair, and to replace. We are going to talk about cell divisions. We identified two types of cell division in our body cells. We have a type that we call mitosis and another type that we identified as meiosis. Mitosis is a type of cell division that takes place in a normal cell body. That is a cell that is not part of the sexual cells or the gametes. While meiosis is a type of cell division that takes place in the sex organs, in the gonads, that is in the testes and in the ovaries. The purpose of mitosis is to produce new cells that replace dead or worn out cells. It also intended to regenerate damaged or sick tissues, while meiosis is intended to produce sex cell that will combine and form the new offspring. We're talking about producing sperms and eggs or ova. Mitosis goes or passes through different stages. These stages will take the cell from phase to phase. Eventually will lead to the production of two daughter cells. If you take a look at the cell here in this model that I'm going to share with you, I want to emphasize that mitosis and meiosis takes place in the nucleus. The nucleus is the center of the activity of meiosis and mitosis, simply because the nucleus contains the genetic materials or the chromosomes that are responsible for the mitosis and meiosis. It does not happen in the cytoplasm. There is one organelle in the cytoplasm that will contribute to the mitosis, and these are the centrioles. These centrioles, when the cells enter mitosis, will migrate to the two equators or the two poles of the cell, and we are going to find out how that acts and how that is used in the process of mitosis or cell division. So the nucleus is the center of activity of mitosis. If you take a look here, the nucleus has a nuclear envelope and it contains the nucleolus, and it does have the chromosomes, which appears here as condensed substance that is not visible. Usually before mitosis, when the cell is in a stage of metabolic activity and growth, but not in an actual cell division, then the chromosomes are not visible. They're just seen as condensed genetic materials, but they're not well differentiated. I'm going to use some models here to show you the phases and the steps of mitosis. The first stage or the first phase of mitosis is the interphase. And the interphase, as I told you, simply the cell or the nucleus has the nuclear envelope still intact. The chromosomes are not visible. When the cell enters the mitotic activity, it goes into the prophase. What happens in the prophase, the nucleus will lose its nuclear envelope, which is no longer visible. It is no nuclear envelope, and the chromosomes will undergo some differentiation. They will be visible. So no nuclear envelope and the chromosomes are visible. The daughter centrioles will move towards the opposite end of the nucleus. As I showed you here, these are the two centrioles. We said each one of these is going to migrate to one end of the nucleus. After that, the third stage is going to be the metaphase. And in the metaphase, as you see here, the chromosomes are lined up. This is simply because the centrioles will migrate and the spindle formation is complete. These are the spindle formations is complete here. And you see the two centrioles and the two poles, or the two ends of the nucleus. Chromosomes will be aligned along the exact equator of the spindles. After that, the cell will move into a different stage. This stage is known as the anaphase. And the anaphase, chromosomes will migrate, will be pulling apart from each other, will split as a centromere and separating the two chromatids. So we are looking into a cell or two cells that there is a project of forming two cells by pulling the two chromosomes apart. And it's important to remember these chromosomes are identical or double. As we said in the beginning, chromosomes will double and line up and then will split at the stage of anaphase. Daughter chromosomes will move gradually towards the opposite pole of the spindle, and they will keep moving and moving, and a split is going to take place here. This is known as cytokinesis. See the split? And we're looking at almost two smaller daughter cells that are being generated or formed. This form or this phase is known as a telophase. At the end of the telophase, there is a complete division, or almost. The cell will almost split, and the two new cells, nucleus, will start to reform and will start to develop their own identity, their own nuclear envelope, and eventually we will have two daughter cells. It is important to say that cells undergo mitosis at different rates, and this depends on the site of the tissues, the type of cells, and the location of the organs. Some tissues regenerate and produce and undergo mitosis faster than the others, like the mucosal lining of the oral cavity, the lining of the gastrointestinal tract, the top layer of the skin, while others have a slow rate of mitosis, like cells of the nervous system, cells of the cardiac tissues. Also, some cells will have a longer time and they do not undergo mitosis. Instead, they die and a new generation will come from the blast cell. Like red blood cells, after 120 days, they get fragile and they get recycled and get dismantled to its basic components, which is protein and heme, and recycled, and a new generation of red blood cells will be produced from the red bone marrow. I want to also emphasize that cells or tissue undergo mitosis to repair, and this repair can take two forms. Some tissues repair by regeneration, and that simply means making new cells that looks similar to the original cells, while some repair by fibrosis, and these are tissues or organs that are hard to repair, like the cardiac tissues, like nervous tissues, and eventually a scar will be formed. And that explains sometimes the fact behind the difficulty in dealing with cardiac or cardiovascular or nervous system diseases. Some of the reasons might be attributed to the fact that these tissues or the cells of these organs lack or do not have the centriole, which leads the process or the mechanism of chromosomes splitting and the stage and initiates the stage of anaphase. Thank you for watching this video, and I hope to see you in another video of our anatomy sessions.